publishing company established by my father, Ted Merrill, physician and environmentalist. His book, which is this book right here, Lessons from Pond Scum, was published the year of his death in 2013. Mr. Mike Fleming, who's behind me somewhere, teaches science and design in the middle years program at Soundview School. Soundview School is an authorized international baccalaureate school here in Washington State. Mr. Fleming read Lessons from Pond Scum when it was first published. Actually, after my family members, he was the first person to get a copy. And he was inspired to have his students pursue an inquiry into making an ecojar. His 7th and 8th graders made jars in the fall of 2014 and observed them until May 2015 when they performed further experimentation. So this year's 8th graders, who made up half of that inquiry group last year, are with us today. And if I do this, maybe you can see that. There we are! Hi! We're going to share with you a little bit about the concept of jars as far as Dr. Merrill presented it in the book, and tell you about the inquiry the students engaged in with Mr. Fleming. And then to finish off, I'd like to tie our thoughts together with the student reflections that you'll be hearing throughout the presentation and the overarching theme that I took from this year's webinars, and then we'll invite questions. The Ecojar project is an easy one to conduct with any age group, as you will learn, if I can get the slides to go, from our presentation. The Ecojar illustrates the concept of the interconnectedness of all living things. Each living thing depends on the others in the jar to survive. Life, as my dad said, wherever you find it, is a dancing, writhing mix of atoms, molecules, and larger clumps and masses, all interactive and interconnected and interdependent. So, how did the Ecojars begin? Over 40 years ago, when my dad and when our family, excuse me, see, I get mixed up if I don't read my notes. Over 40 years ago, when my father, Dr. Ted Merrill, quit teaching biology to return to medical practice, he had a saltwater aquarium in his lab. And he had created that with samples from tide pools that we gathered on our camping trips to Maine. He couldn't bring himself to throw it all away, though, especially the critters, so he tried an experiment. He scooped some sand and mud from the bottom of the tank into a clean glass jar and then filled it almost to the top with water. He added some of the critters along with some algae. The jar was sealed with paraffin and set in a sunny window. The jar's appearance changed over time, but when we moved away 3 years later, my dad left the jar behind in the windowsill, and there was still one snail living along with some green algae. Here is a photo of Dr. Merrill's last jar, and he had jars on his windowsills for most of those 40 years after he made the first one. So this jar was decommissioned shortly after his death 3 years ago, unless it's still, I don't know, other family members may actually have it. One of the things that he proposes in his ideas is that the Ecojar is like Earth. And we'll talk about that in a minute. But I have to indulge myself and show you this poster. This is the poster that was hanging in his study while he was writing this book. Though he was a physician for most of his adult life, he was always an ecologist. He felt that this book, this message about the critical need for care and critical need to care for Earth and its life systems was his most important book, and he wrote several, but this is the one that he felt was the most important message he had to share with the world. And the overwhelming message of all the webinars we've watched this month for me is the same. Take care of the system that supports the organism, and you take care of the organism. So that, to me, is pretty big deal. Let's see. Okay, here are the concepts that he shared about how the Ecojar is like Earth. And if you have the Making an Ecojar brochure, can you see that there? I have to ask Deb how, and maybe she'll tell my helper here, how to share that document with you. I've uploaded it, but I can't figure out how to put the link into the stream. But You got it with your registration materials too. Okay, awesome. She's going to do that for you. So Earth, the whole recap of this set of concepts is in this brochure. So there are some bullet points though. Here's what Lessons from Scum has to say. The Earth is a spaceship in the largest sense of the word, a closed ecological system supporting a highly diverse variety of life forms, all interdependent and self-balancing, and driven entirely by energy from the sun. Except for its scale of both size and diversity, he felt that the Ecojar is an authentic model of the Earth, but it's small enough to fit more easily into our minds. By careful comparison, we can confirm its similarity to the Earth's system. We may not usually think of our planet this way, but the similarity is undeniable when Earth is viewed from a distance. And I'm going to talk about that a little bit at the very end, about Earth viewed from a distance. So Mr. Fleming is going to get to talk next. How did Soundview students come to make the Ecojars? I'm going to pop out of the screen.
Hello, everyone.
Make sure they can see me.
Okay. Can everybody see me okay? This is my first webinar.
That's all of our first webinar.
So I've been asked to talk a little bit about how the Ecojars came to be of use in our classroom. In the 7th and 8th grade rotation here at Soundview School, we do 1 year of biology. And the final unit of the biology curriculum is ecology. And when Ms. Stein brought this project to me, she just thought I would find it interesting. It sounded like a great fit for that particular unit of study. So because we could make the jars and then continuously make observations on them over the year and then discuss them, do further analysis when the ecology unit came up in the spring. So just the basic progression here, we created the jars in the fall in order to study them over the course of the year. After creating the jars, we recorded observations first at weekly intervals. We noticed that a lot of changes were happening in a very short time frame after the initial creation of the jars. The emphasis in those observations was on all about observing the different types of organisms that we saw and how they were changing, how the numbers of them were changing, were any of them increasing, did it look like any of them were dying off. And then at the conclusion of the activity, after we had observed them over the course of the year, we opened up the jars and did a little bit of further analysis. Took some samples of water, looked at them under the microscope. We tried to characterize the aromas that came out of the jar. And when Ms. Stein did this before, there were some very, very funky ones. That was not the case with us, though. It was I was prepared for some really bad smells, but it just smelled like pond water. And then we kind of concluded and wrapped up there about looking at the connections between the different types of organisms that were in there. And I think now we'll turn this back to you.
Yep.
All right.
Hey. All right. So there were some challenges in creating the jars, and we wanted to be sure that we met all these challenges. One of my main concerns was to find a way to create 20 jars without negatively impacting the ecosystem we would visit. And in late summer of 2014, I found a fantastic spot with a big area of stagnant water adjacent to a stream not too far from school. So in October, we took a field trip to fill our jars. And by October, temperatures had cooled dramatically, and deciduous leaf fall had completely covered this stagnant area. So even small puddles of water and areas with mud in them were filled with leaf litter, and all of the green algae and plant material were gone. That was a problem. Let's see, here's Mr. Fleming preparing the group before we start. That was a big group. all of us trooping down the trail, and I got lost going. I took a wrong path the first time I went back to the site because I had directed it. I had gone to it from a different direction. Here we all are gathered at the stream right before we found out there wasn't any green stuff to be had. So all we had was a running stream. What should we do? Well, rather than abandon the experiments altogether, we just changed the focus of the exploration. Mr. Fleming had half the students take water and soil from faster moving water in the stream proper and half from a slower part of the stream. They documented location and type of material. Now you just get to see pictures of kids in water, and that's pretty fun. So observing the area, they were making notes about the area before they went down to the water. There they are actually in the stream. They were in groups collecting their material and their water, and then they were making notes in their lab notebooks. And it was a big group, but there's somebody, can you? Yeah, that would be interesting. So there are a lot of things alive that you just can't see, especially in water. There's all the like worms, like we talked about in water. Noah stream, one of our favorite critters. Okay, so we had water, we had soil. Mr. Fleming and I went on our own field trips and collected green algae and other plant material from 2 local lakes. The lakes were about 20 miles apart. What was the plant that you found at Green Lake? Do you remember that?
Primarily Elodia.
Elodia.
Green Lake is kind of overrun with that.
Okay, and I was at Serene Lake, and it was just green, floaty stuff. And so we got as big a jar as we could, filled it with green material, and then the kids they had their jars. The jars needed to settle overnight to protect the green matter from being silted over. If you put your green material in too soon, the dirt that's floating in the water will silt over the green matter, and then it won't work the way it's supposed to keep your jar running smoothly. Okay. The jars needed to settle overnight, and then there they were, settled. They look pretty good. And then the students added the green plant matter to each jar, and then they were sealed using toilet sealing wax. What was that like, you guys? I understand it was a very sticky process. Yes. Oops, I'm going to throw my slides all over. So next we'll talk about observing your jar, because as you can see, all you need, if you have the brochure or if you've listened to us talk, you need water, you need some dirt or mud or sand from the water that you're observing, and you need some green material. My dad and I made saltwater ones. My husband and I made a great big saltwater one when we were first married. But you can use a stream, a pond, a roadside puddle, whatever you can find that you can include some green material in. Well, we'll talk about that a little later too. You can make an Ecojar. So how does observing your jar work? And it's fascinating to me that Mr. Fleming's idea was always to observe what had been happening by opening and examining the jar. And we've had lots of conversations about how to examine the water and what's in it without unsealing the system. Because my dad was all about, here's the sealed system. It's a model of the earth, and that's what it's supposed to be. So here's what my dad says, and then Alex is going to share her observations about the jar. Every jar is different, and yours will produce different results and lessons that I have never encountered. That's what my dad said. That's what Dr. Merrill said. And that's really true, and that's what I think we'll learn from these guys. So here's Alex.
Hi. Okay, so I was at Fleming last year, So my observations were very different because I wasn't very clear on how to take observations. So, but here are my thoughts. So life is very difficult to sustain, but if you leave it alone, it can flourish. And that's kind of different from how I've been raised, because you have to take care of things, like animals. You have to take care of them. You have to feed them. You have to water them, stuff like that. But the Ecojars themselves, they were a difficult project, and it was very unclear if there was a life inside of them, even though there was. This project was one of patience, waiting to see if our Ecojars would sustain life, and if we could tell if there was life in them. Mostly, they were brown and dead looking all the time. But when we went to create the Ecojars, none of us kind of were told what we were doing. We were told, oh, we'll talk about it later in the year. So we were unclear on what we would be doing. So we went and we jumped down to the pond and scooped some dirt. If I were to do this project again, I would place the jar in a more light source focused environment. We placed ours on a windowsill where there was light, there wasn't much of it. Just because I am curious to see that if mine would have flourished. Mine was very brown and dead looking the entire project, though I did have worms and water bears. I'd also take more observations, because we only looked at them every 3 months, and the course of 3 months is a long span of time, but we weren't looking at them. So we'd go back and see what we wrote. Oh, it's the same thing over and over and over again. If we took more detailed notes, we would have seen, oh, this is how it's going. This is what has changed over time. Yeah.
Great. Thank you. Can you hand that to Bridget? And as my dad says right there for you on the screen, be patient. Most changes are very slow. Okay, observing the jars. And I say anyone can do this. I used my own observation notes. I made a jar at the same time as the kids, kind of with the leftovers. So I didn't have a lot of green stuff. And I made the mistake of using a canning jar. And a canning jar has lots of texture along the sides, and so a lot of little green stuff grew, and the glass distorted, and I couldn't see. But you can see from this picture that I did discover many, many, many organisms. I had hydra, and I had this weird segmented creature that was very active, and he was almost ½ an inch long, or 2 of those. They ran around. Some of the little sand shrimp dudes, the scud or whatever they were, they had babies. I mean, there were egg cases and like eggs in their bellies, and it was pretty amazing. So observing the jar, anybody can do this. Get a good clear jar, make it a glass jar, seal it. The first time we did it here at school, I used a pickle jar and painter's tape, and it didn't seal very well, and then the library smelled really bad, but we'll talk about that in a moment. All right, here are the jars that the students made, and I photographed them after they'd been in this windowsill for about 4 months. Now, my dad would have said this is ample light. It's true that some jars were behind others, so they may have gotten a little bit less light. Interesting colors of algae are beginning to form in these jars. Some look dead, but every jar had life in it, as far as I understand. Okay? So Bridget is up next. She's going to talk a little bit about her observations. It's weird talking to nobody. You can say it. It's okay.
People, lots of people. Okay, I'm going to read my kind of reflection for what I learned from the project first. So what I wrote was, life exists on many small levels that require more investigation to see. Even when it seems like there's nothing at all, life finds a way to prosper in incredible ways. This project showed us that it is possible for life to find life in places you didn't think were possible before. So I know Alex said that hers looked really dead and brown. Some people's looked more green, like Ms. Stein was saying. The light was different in some areas because they were behind each other, so it just kind of varied, which we learned later on. And yeah, so we all saw a lot of different things. So it was fun when we first made our jars to kind of compare each other's and say, I have a worm in here, I can see moving, stuff like that. Yeah, a lot of ours had critters in it and stuff like that, different algae. And then we found worms and named them. I don't know if you guys remember that. We named all the worms in our jars. Yeah, so it was really fun to go get the stuff to put into the jars. Kind of strange, like Alex said, we had no idea what was really happening. We kind of vague on what the whole deal was. So we were sort of, just kind of going with it. Yeah, so it shows kind of how resilient life was, like even when it seemed like it was really, there's nothing there at all. Looking deeper showed us that there were some small critters there and algae growing, things like that. Yeah. So I was saying that if I was to kind of change one thing, it'd be to take really good observations, because I know sometimes mine were really brief and short. So if I was to do this again, I would definitely write detailed observations and draw pictures, things like that. So when we first started doing the project, we checked in more frequently, and we could see drastic changes like the water settling, things like that. And also, again, the ceiling wax was a mess. So pro tip, don't use too much of the wax or it'll get everywhere on your hands and clothes. Yeah, that's pretty much what I wrote.
Thank you for that. Okay, great. Thanks, Bridget. How's our time? Good. That's exactly what I was hoping for. Okay. The jars were arrayed in the, let me see if we go, yep, in the most consistently sunny window in the middle school building during the months of the inquiry. And this really was plenty of light for the inquiry like we talked about. So a few years earlier, I made some jars in the library when this book was being written, and they did not have enough light, just enough light from a window in a door, and those jars rotted. Their appearance was very different from these, kind of weird, blue-green, strange, and they smelled. And I'll talk about the inquiry that we did about those later, but Emma is now going to talk about what she observed. I'm just going to hop up and down in this little dude. That's great.
Thing. You guys are doing great. Okay, well, hi. So, well, when we did this project, I think we've talked about this a little bit already, but the concept that we kind of hit on the most was that life can come back even when it seems like that it is all gone. There can still be enough life to keep the ecosystem going. So, for instance, you don't have to throw away all the water in your dead goldfish's bowl. Yeah. So, yeah, that was interesting. So there are a lot of things alive that you just can't see, especially in water. There's all the like worms, like we talked about, and water bears. These little things that rely on the ecosystem around them. So I think that's what we were really studying, and I really like that part. If I was to do this project again, though, I would probably get my water and my dirt, a weird thing, from a more active ecosystem with more plants. Like Mrs. Stein pointed out, the ecosystem where we got the water from and the mud from didn't actually have much life in it anymore. And we even had to get the algae from a different source. I don't know if that affected it or not, but I mean, although the water that we did get it from had interesting creatures in it, for the purpose of the experiment, I think that it would be beneficial to have more creatures in the jar, just to see how it worked out, or to study more in depth the creatures, and you could see a wider variety. I think one of the questions was, what did you name worms? We were really heavy into our Percy Jackson phase right then. So we had like Frank, Hazel, Percy, and Annabeth. Yeah.
That question was from Miss X.
And that makes Mrs. Stein, the librarian, happy that you named the first character from a book. What can I
Yeah.
Who says awesome names, you guys?
Yeah. Okay. All right. Thanks, Emma. I think Emma's point about a more rich system is a really good one. And my dad often used ponds, a pond or a large puddle. And I was worried about adding plant material from different sources, but he did that too. If he didn't have enough of something somewhere, he did more. And my husband and I made a huge one in an old glass carboy. And so we had, you know, an area of sand that was about this big, and we had several snails, we had sandworms, and that thing lived until it froze outside one year. So it was a very successful jar. Here is one of my favorite pictures because it shows a sampling of the growth patterns in all the different jars. I mean, that's not all of them. There were 20, but that's 4 or 5 of them anyway. That just shows you the differences in these jars over the 4 months. Now, remember, some had the Erodia, did I say it right? Did I miss that? And some had just this soft green algae that I got out of Serene Lake. And Serene Lake was just, no, Martha Lake, sorry, was just sandy bottom with some green stuff floating. There was very little other vegetation. So the jars, I think, were affected, as Emma wondered, by the material that was added to them, not from their own source. So Aubrey is going to share with us all now.
Surprise, I'm a boy. I didn't write that much because I'm lazy. If you look in the top right corner, you can see my Ecojar, the one with David. There's some goes on in there. I wrote that basically what everybody else wrote. There was life in the jar basically the entire time, just because the plants looked like they were dead. There were still microscopic organisms that you could find under a microscope, obviously, which brought the plants back to life sometimes. So you could see more green, and sometimes the water just turned cloudy and crazy.
Yeah, thank you. Right. Turning my pages here. So this picture was shared with me. 8th graders, is this exhibition night? There is signs that say do not touch. Okay, all right. That's great. So the school has an exhibition night, families come, and so the kids got to talk a little bit about the jars, and everybody could see the jars, and it was fun to hear people talking about them afterward. I didn't get to go to that exhibition night, but it's fun to share with the community. Lily is up next.
Thank you guys. This is great. It's going great. I've watched a lot of webinars and it's been nice.
Hello. My name's Lily, and I was in STEM last year, so I did this project along with past year's this grade. So the Ecojar, or Ecojars, provides a way to view a small representation of our own ecosystem closer up. At first glance, life seems absent in the small, still jars. But if left alone with the proper sunlight conditions, you can observe signs of life flourishing in its own little world. The Ecojar is a microcosm of our own ecosystem. Vocabulary. With the proper care, sources, and absence of contaminants, we would be living in a flourishing world. So if I were to repeat this project, I think I would collect my water from a different part of the pond. I collected it in an area that didn't look like it had a lot of living small things. So I would collect it in a section that has more active water, microinvertebrate, and And I do have to say that we collected our water from a stream, and that was not our original intent. I chose an area of stagnant water that wasn't there anymore when we went to get our sample. So that's one thing I agree that all that stuff is important. Where would you collect your samples? After creating the jars, says Mr. Fleming, students recorded observations at weekly intervals, and as the year progressed, these shifted to a monthly schedule. So here are some of the students making observations on their jars. And Julia is going to share with us.
I'm Julia. So as the rest of the class said, that while we were making observations by just looking at the jar, it didn't really look that we had any plants or not animals or microinvertebrates in the jar. But eventually, when we took pieces of plants out and looked at them under microscopes, I did find like 2. So don't give up and keep looking. I was slightly disappointed that I didn't find very many macroinvertebrates. So if I were to do this again, I would pay more attention to where I collected my water and dirt from, even though the conditions of the stream weren't very good and there weren't very many macroinvertebrates or plants in the stream. And so just pay attention to where you get your water.
I really do think it has a lot to do with the time of year that we collected, because in July everything in the area was thriving, and I know that in the summer, even a month before I went to the lake to get the green stuff I got, people said it was just full. They'd walk out and up to their knees in green goo. So you guys have mentioned already what kinds of organisms you were finding, some tardigrades, water bears, worms. Did you see any segmented creatures? Do you remember? Yes. Mr. Fleming says yes. I was just blown away at how much stuff was in my little jar. Okay, and now Winnie is going to share with us. We just have a couple more students and then we'll wrap up. All right.
Hi. So the Ecojars showed that even when life seemed dead or still, there's still ecosystem developing with enough sunlight. So pretty much what most people say. If I were to do this project again, I would collect the elements of my Ecojar from a source that includes more plants and macroinvertebrates. Performing this experiment, I thought we didn't pay much attention to what was happening in the jars that we used. So sampling the water every month or so under the microscope would be a huge improvement.
Okay, and as I said, my dad was seal the jar and leave it. So the idea of opening the jar and sampling to see what's in it is just fascinating to me, to see how we can change the way we look at the inquiry. Okay, these guys were sampling. This was after 7 months, and I see that I left one of the things. Some of the things observed were note and didn't write anything, but that's okay. You've heard what the students observed. So during the ecology unit in the spring, the jars were opened and the material was examined under the microscope. Mr. Fleming, as he said earlier, said there was no odor really other than that kind of pond water smell that you get. And we did try examining jars. The jars that we did in the library, they had this sort of flocking over everything, and it was a deep blue-green, which I've seen in my jar now, and dark, kind of tarry stuff around, and it smelled hideous. Some kids just couldn't even stay in the room, but some did persevere and look at stuff through the microscope, and it was astonishing how much was in the jars even when they hadn't even been in the light. Because after they got stinky, I took them out of the area where we spent most of our time. All right, Mia is up next.
Hi, I'm Mia. Something I took away from this project is that life is able to grow if it has the right conditions, even in a sealed glass jar, and it seems like I won't be able to. If I could do this project again, I would pay attention to the variables that went into the jar, and where I took the water from, and the exposure to sunlight and stuff like that. Have faith in your jar, because even though it looks brown and dead, there's huge life in it, probably. And I think everybody had a little bit of life in the jar. So.
Cool. Thank you. Okay, and remember that you can record your questions any time in here, and we'll get to them at the end. These are the control jars that Mr. Fleming collected, to which no additional green matter was added. These were photographed last month. There's still living stuff in there. And there's some green stuff going in there. So yes, as Mia said, have faith in your jar. There's stuff happening. It's amazing to me how much is still growing. Okay, we're going to tie up a few things and then we'll get your questions. This is really the key. When we started preparing this presentation, our focus was mainly on the inquiry and telling you all how it came about and what it looked like. But there really is more to it than just that. The overwhelming message from all the webinars that I've been able to watch this month has to do with that system again and how we protect the species within a system. So can you think of one concrete way that you can protect a system near you? That's a question that I would have. And now we're going to go crazy with. Okay, so be thinking about that. Oh, my feed reader is madly writing here. And I'm going to just go to the next slide. Here's what my dad had to say in Lessons from Scum. And it's about questions. Each time you find the answer to a question, it unveils more questions, which I noticed from what the students were saying. As your accumulated knowledge grows, the surrounding circle of questions, of mysteries, grows larger, deeper, and richer, a depth and expansive mystery without limits. So if you do an Ecojar once, and you find out some stuff, and you do it again, chances are you'll find out some other stuff. So now we're going to open it up for questions. I have one more page, and I'll talk a little bit about the for further inquiry stuff, but we'll just see if there are any other questions. Okay, Emma's going to answer this question that somebody had. So say what the question is, where it came from.
Okay. So the question was, are there no holes in the top of the jar? And it was from
The red school.
The red school. Sorry. And no, there were no holes in the top of the jar. We sealed it with toilet wax really, really well. It was really disgusting. Got all over your fingers. It was impossible to get off. And I was surprised at how little air there was in the jar, but you did have to leave some. You didn't fill it all the way to the top with water. So.
Okay, thank you. That was a good question. The idea of a sealed system is hard for us to consider. Is there another question? Do you see any more questions? I have to let these young eyes read the chat stream because my bifocals don't work, so I'm leaning over so she can see. Any more questions? I'll go ahead and go to the slide for further resources, and we can check back on questions in a minute. Guess it doesn't affect you. So, of course, I would say read the book. You can get it both in print and as an e-book. My dad was very proud of this book, as am I. Another thing I would mention for further inquiry is a really cool thing I ran across when I was researching just about the Earth. And actually, I was looking for images of the Earth, and I found a documentary called The Overview Effect. And it's just a 20-minute video, but it shares the experience of astronauts and their reaction to the importance of saving the Earth after they saw it from space. Very moving, very interesting to experience, and I recommend it highly to anyone. It was life-changing for these astronauts to realize what a small ball we live on, I think, and what a fragile place. Another thing that I would recommend is a book called Harmony, A New Way of Looking at Our World by His Royal Highness Prince Charles. This book, I think, is out of print. I got it several years ago. You can find it on Better World Books, and I think Amazon has some used copies too. Really a great book. My dad read it, loved it, mentioned it in this book. Another book that I used with the 8th graders when we made our stinky Ecojars is called Thinking in Systems, a Primer by Donella H. Meadows, and it was a book that really informed a lot of what my dad's thinking was too as he wrote this book. I used some of those lessons, and they were very accessible to even to 8th graders. And then the last thing is a PDF publication. It was a very helpful resource for identifying some of the macroinvertebrates, and it's the Stroud Water Research Guide to Identifying Freshwater Macroinvertebrates. And I loved it. It's downloadable as a PDF as well. Again, I think Deb shared the the link to the brochure. So you can use the brochure. You can also email us anytime. I'm going to put the email in here.
I think this is actually a question for you.
Okay.
It says, will this bibliography be available later, or the webinar as a whole? This Steph G says, they missed the first.
Yes,
couple
okay.
minutes.
The webinar will be available on the YouTube channel of the National Biodiversity Teach-In, which is very exciting. They've made all of the webinars available on their YouTube channel. And I am going to make this, I meant to put this in the documents too, Deb. I'm sorry I didn't do that. This list of links. But you can email me. Now let's hope I spell it right. Make sure. Yes, books at homeostasispress.com. You can email me and I will send you a copy of these materials. So, are there any other questions?
Was there another one?
Okay, Alex looks to be ready to answer a question.
I'm back. Okay, so this question came from Ryan, and it's asking, what do you do with it when it's done?
Yeah, what
The jar. The jar. So, it's never done. Let's say it that. When the experiment is over, the 7th grade at the time, took them out to our forest, unsealed them, got very sticky, and dumped them out. The plants and all the animals like stuck to the jar, so you had to run back inside, fill it up with water, swish it around, dump it back out. Swish it with water, dump it back out. It took at least 7 times for it to be clean, and we had to get the toilet wax off. And that was a sticky process, and it took an hour and a half to get like everything clean and done. Yeah.
Okay. And I've never been able to bear to dump out a jar. Having looked at some of the little critters through the magnifying glass and under the microscope, I just can't do it. So I even have through the magnifying glass and under the microscope, I just can't do it. So I even have the rotten jars in the sunroom in a corner at home. Okay, good. Looks like people are asking questions. This is great. Great job, middle schoolers. That's what Miss Kay says. So they're very impressed with you. I've watched a lot of them. You guys did a good job. Good job. Okay. Anything else? We'll just wait. Okay. Really?
Elgin says we're really impressed with your presentation. We want to know.
Oh, awesome! 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 I want to see pictures when you do. These guys do a lot of stream science. So they monitor the stream in their area.
Fourth graders.
Yeah. Oh, the 4th graders too. Oh, that's so exciting. Yay. Okay. Yes, do it in the summer. I think that's really the key to the success. We wanted the jars in October because Mr. Fleming really wanted the students to observe over a long period of time. So that was the key, is that we just didn't have the right time of year for gathering the material. And a pond, after this fall, I found a couple of great ponds that are not far away. So yes, you have to decide if your jar is done. And oh, hi, Ani! I'm so glad you're there too. That's excellent. There were some people on the planning committee with me that joined today, so I'm very honored that everybody took their time. We just want to thank everyone, all 700 and however many of you who came. Can you write down the list of
I will, but the Red Schoolhouse says this is our favorite webinar.
Ah! Yay! That's really awesome. Thank you so much. We're just really
Does anybody else
appreciative.
have any questions?
All right. We got one person typing. Looks like Miss Pearman is typing. Oh, Miss Pearman, who's the organizer? She likes ours the best too. Oh, there you go. I don't normally turn my head away from the camera, but there are all these kids behind us. So there you go. Well, we've just had a really great time communicating with everybody, and we're madly writing down all the schools because we want to know all the places people came from, where you're all from. And some people or other eighth graders. Oh, other students are coming back to school, so it's easier. Thank you so much. We just really appreciate your time. I'm going to go back because I know you can get to this slide. I'm going to go back to slide number 1, 2. Just going to go back to a picture of the jars because I think.
Carla A says, I work in a large wetland area and we will try this with our summer camp participants.
Oh, awesome. The saddest thing is knowing that if you're working with a student population that's not permanent, like can you keep jars for a long period of time? And I really was worried about the ecosystem and what might happen if we collected a lot. But look, this has just been an amazing inquiry and a really great exploration of the system. So I think Soundview School needs more than one copy in the library too. That's the other thing. So, awesome. Okay, what's the math?
I'm interested, from Steph G, I'm interested in scaling this project up for college students. Any thoughts?
Well, my dad did it with college students. I mean, he taught college students biology, and so it just depends on what your interest might be. Mr. Fleming wanted to put one-way diaphragm in the jar so you could take samples without unsealing it. So if you could figure out a way to do, membrane that you could use a syringe or some for drawing material from the jar without losing its seal to keep that sealed, that's one thought. And you could do control jars that aren't opened and jars that are opened. That's another way. Just depends on what scale, but be prepared for it to take a while because they take a while to observe. I guess that's the key. My dad had some he observed for, I think, I want to say 7 years. Some jars he kept for that long and observed regularly for 7 years. So I don't know if that works. Oh, I'm sorry. No, you don't have to be sorry. You're doing a great job. Okay. One more. Did I write 12 years? I didn't write 12 years. That's funny. There's 2. Oh, I wonder if one is. No, one shouldn't be. Well, my husband is logged in too, but he's not logged in as me. So, interesting. Maybe that is me. Him. Yep. Whoever I am is writing yes.
Yes, he is.
That's my husband writing it. 12 years. He says 12. Awesome. So they can go for a long time. So you could use a jar that you have for observations over a long period of time and then make jars that are sampled from too. I think that's great. So our lovely chat stream monitor has written down all the schools that participated and all the individuals, and we just want to thank you so much. Are there any further questions? All the kids are getting up to see how many different schools were involved in our webinar today. Okay. All right, I think
You guys can pick it up.
we are done. I'm going to get out of the way and let the kids just all wave to you one more time. Say thank you.
Hail Satan 666.
Hey, Aubrey. Really?
Yes, really.
It's just the people kind of right here.
Yeah.
People are typing to you guys.
Aubrey.
Guys, no, Emma, go down.